Blood types are one of the most fascinating and essential aspects of human biology, playing a critical role in medical procedures, transfusions, and even our understanding of human evolution. The existence of different blood types is a result of genetic variation, and these variations have significant implications for health, disease susceptibility, and compatibility between individuals. But why do we have different blood types? What purpose do they serve, and how did they evolve? To answer these questions, we must delve into the science of blood, the genetics behind blood types, and the evolutionary forces that have shaped this diversity. The concept of blood types was first discovered in the early 20th century by Austrian scientist Karl Landsteiner, who identified the ABO blood group system. This system classifies blood into four main types, A, B, AB, and O. These blood types are determined by the presence or absence of specific antigens on the surface of red blood cells. Antigens are molecules that can trigger an immune response, and in the case of blood types, they are sugars or proteins that are attached to the surface of red blood cells. The ABO system is based on two antigens, A and B. A blood type A has the A antigen, blood type B has the B antigen, blood type AB has both antigens, and blood type O has neither. In addition to the ABO system, there is another important blood group system called the Rh system, which determines whether a person is Rh positive or Rh negative based on the presence or absence of the Rh antigen. The differences in blood types are determined by variations in the ABO gene, which is located on chromosome 9. This gene encodes an enzyme called glycosyltransferase, which adds specific sugars to the surface of red blood cells to form the A and B antigens. The A and B alleles of the ABO gene produce slightly different versions of the enzyme, leading to the formation of different antigens. The O allele, on the other hand, produces a non-functional enzyme, resulting in the absence of A or B antigens. The combination of alleles inherited from both parents determines an individual's blood type. For example, if a person inherits an A allele from one parent and an O allele from the other, they will have blood type A. If they inherit a B allele from one parent and an O allele from the other, they will have blood type B. If they inherit both an A and a B allele, they will have blood type AB, and if they inherit two O alleles, they will have blood type O. The Rh system is determined by the presence or absence of the Rhd antigen, which is encoded by the Rhd gene. Individuals who have the Rhd antigen are Rh positive, while those who lack it are Rh negative. The Rh system is particularly important in pregnancy, as incompatibility between an Rh negative mother and an Rh positive baby can lead to a condition called hemolytic disease of the newborn, where the mother's immune system attacks the baby's red blood cells. The existence of different blood types has significant implications for blood transfusions and organ transplants. If a person receives blood from a donor with an incompatible blood type, their immune system may recognize the foreign antigens as a threat and mount an immune response, leading to potentially life-threatening complications. For example, a person with blood type A cannot receive blood from a donor with blood type B, as their immune system will produce antibodies against the B antigen. Similarly, a person with blood type O can only receive blood from another type O donor, as their immune system will produce antibodies against both A and B antigens. On the other hand, individuals with blood type AB are considered universal recipients, as they can receive blood from any blood type without triggering an immune response. But why do these different blood types exist in the first place? What evolutionary advantage, if any, do they provide? One theory is that the diversity of blood types is a result of balancing selection, a process in which multiple alleles are maintained in a population because they provide different advantages in different environments. For example, some studies have suggested that blood type O may provide protection against severe malaria, a disease caused by the Plasmodium parasite. Malaria is a major selective pressure in many parts of the world, particularly in Africa, where the prevalence of blood type O is higher. The Plasmodium parasite infects red blood cells, and individuals with blood type O may be less susceptible to severe forms of the disease because their red blood cells are less likely to form rosettes, clusters of infected and uninfected cells that can block blood vessels and cause severe complications. On the other hand, blood types A and B may provide protection against other infectious diseases. For example, some studies have suggested that blood type A may be associated with a lower risk of smallpox, while blood type B may be associated with a lower risk of certain bacterial infections. These findings suggest that the diversity of blood types may have evolved as a way to protect against a range of pathogens, with different blood types providing advantages in different environments or against different diseases. Another theory is that blood types may have evolved as a way to regulate the gut microbiome, the community of microorganisms that live in the digestive tract. The A and B antigens are not only found on red blood cells but also on the surface of cells in the digestive tract and in bodily fluids such as saliva. These antigens can influence the composition of the gut microbiome by serving as a food source for certain bacteria or by preventing the attachment of harmful bacteria. For example, some studies have suggested that individuals with blood type O may have a higher risk of developing peptic ulcers because they lack the A and B antigens that can protect against the Helicobacter pylori bacteria, a major cause of ulcers. The distribution of blood types varies widely across different populations, reflecting the complex interplay of genetic, evolutionary, and environmental factors. For example, blood type O is the most common blood type worldwide, particularly in the Americas and Africa, while blood type A is more common in Europe and parts of Asia. Blood type B is relatively rare in most populations but is more common in Central Asia and parts of India. Blood type AB is the rarest blood type worldwide. These patterns of distribution are thought to reflect the migration and adaptation of human populations over thousands of years, as well as the impact of infectious diseases and other selective pressures. In addition to the ABO and Rh systems, there are many other blood group systems that have been identified, each with its own set of antigens and genetic variations. These include the Kell, Duffy, Kidd, and MNS systems, among others. While these systems are less well known than the ABO and Rh systems, they can also have important implications for blood transfusions in pregnancy. For example, the Kell system is particularly important in prenatal testing, as incompatibility between a mother and baby can lead to hemolytic disease of the newborn. The study of blood types has also provided valuable insights into human history and migration. By analyzing the distribution of blood types in different populations, scientists can trace the movements of ancient human populations and their interactions with each other. For example, the high prevalence of blood type B in Central Asia is thought to reflect the influence of the Mongol Empire, which spread this blood type through conquest and migration. Similarly, the distribution of blood type O in the Americas is thought to reflect the genetic legacy of the first human settlers who crossed the Bering Land Bridge from Asia tens of thousands of years ago. In conclusion, the existence of different blood types is a result of genetic variation and evolutionary forces that have shaped human populations over thousands of years. The ABO and Rh systems are the most well-known and clinically important blood group systems, but there are many others that contribute to the diversity of human blood types. These differences have significant implications for health, disease susceptibility, and medical procedures such as blood transfusions and organ transplants. The evolutionary advantages of different blood types are still being studied, but they likely reflect the complex interplay of genetic, environmental, and infectious disease factors. By understanding the science behind blood types, we can gain valuable insights into human biology, history, and the ongoing challenges of medicine and health.